I'm a postdoc in Dagan Segal's lab at UT Southwestern, and I'm really interested in studying mechanisms of cancer cell adaptation. And here we go. And when we think about what drives a phenotype in cancer, a lot of times we think about changes in the genome or the transcriptome which drive a phenotype. But there's a whole other layer of regulation in terms of at the cell biological levels, in terms of cytoskeletal dynamics, membrane dynamics, morphological motifs that also drive a phenotype. And I would argue that these cell biological processes are really the first responders to environmental cues at short time scales, which later feed back and regulate these other processes upstream. And I think our insight into these processes is limited because of the difficulty of observing these types of phenotypes in situ in complex environments. So that brings me back to my title slide, where I want to talk about insights from my postdoc work using zebrafish and 3D culture models to study cancer cell adaptation. And I've included here in my title slide as an example these 3D renderings of human cancer cells imaged in a zebrafish and pseudo-colored according to their cell shape. And immediately you can appreciate there is the large diversity of cell shapes we see. And since form often follows function in biology, it's quite possible that these different cell shapes represent different signaling states, which would contribute differently to disease progression in cancer. So cell morphology is really emerging as an increasingly quantitative readout of cell state in systems biology studies. For example, this Schwartz lab paper that talks about how different cell morphotypes correspond to different transcriptional signatures and different in vivo functions, or this beautiful work by the Carpenter lab using their cell painting morphology classification system as a way to distinguish cellular response to different chemical drug perturbations. And because morphology is such an accessible, quantifiable readout, many rely on it as a functional output, especially in studies that look at different microenvironmental conditions, because the morphology is one of the first things to change in these systems. And it's quite common to see different morphology emerge from different 3D substrates in 3D culture systems. And we can only assume that's also the case in a more complex tissue where we have a much higher diversity of chemical and mechanical cues, but observing these aspects of cell adaptation at a single cell level in a tissue is difficult to do due to challenges in imaging. And that's really where the zebrafish model comes in as an ideal model to study these aspects of cell adaptation because of its combined advantages of accessibility to high-resolution imaging, the throughput necessary to capture true heterogeneity of data, and the complex microenvironments of a vertebrate organism. And we can compare the different seeding sites within a fish as very distinct microenvironments. For example, this hindbrain ventricle, which is this fluid-filled compartment that's very rich in hormones. The paravitalin space, which is this avascular region sandwiched between two membranes, and the caudal hematopoietic tissue, which is this area that's very rich in vasculature, which actually serves as a stem cell niche. And the advantages of the zebrafish are especially leveraged when combined with innovations in an imaging modality called light sheet microscopy. So what's special about light sheet microscopy is, as opposed to confocal microscopes, which are very good at creating optical sections of a 3D sample, but illuminate the whole sample in doing so, a light sheet microscope selectively illuminates only the plane of interest. And this has the combined advantages of comparable optical sectioning, but massively reduced phototoxicity for your sample and massively orders of magnitude higher speed of acquisition, which also enables higher throughput of imaging. So that makes light sheet microscopy especially amenable to imaging very fast 3D processes in biology. For example, this immune cell attacking this cancer cell. And simultaneously, because of the low phototoxicity, we're able to image over very long time scales. For example, in this developing zebrafish embryo over many, many hours. So in my postdoc, I focused specifically on a pediatric cancer called Ewing sarcoma as my specific model to study non-genetic adaptation. And Ewing sarcoma is a pediatric cancer of bone and soft tissue, and it's driven by a single fusion oncogene of two transcription factors. And it has very variable patient outcome. About 70% of patients have a very treatable localized disease, but for the 40% of patients who develop metastases, we have very meager prognosis, and we really have an urgent need for a better clinical understanding of disease progression here. So something that's characteristic of Ewing sarcoma and actually many pediatric cancers is a very, very low mutational burden, especially when you compare that to many adult cancers. So this combination of very low mutational burden and these differences in patient outcomes suggest that environmental effects may drive the distinct patient outcomes in this case, is making Ewing sarcoma a particularly good model to study these non-genetic mechanisms of adaptation. So in my postdoc, I leveraged the advantages of the zebrafish and high-resolution imaging when I developed an assay using xenografts in zebrafish to study plasticity in Ewing sarcoma. So what I did was I injected mixtures of fluorescently labeled cancer cells into different seeding sites in the fish, and then imaged those with a high-resolution microscope, where I was able to extract from the images 3D volumes of single cells within a cluster of cells within different seeding sites in the fish. And then from those 3D volumes, we could do just some simple geometric feature-based quantification of the different cell states to find differences between the cell morphologies. And this work was a collaboration with Hanya, who developed the computational pipeline, and Bo Ray, who developed some of the optics. And it's been a really fun project. And it's actually proved quite fruitful in finding differences in cell states as a function of various aspects of interest. For example, we could ask how the different tissue microenvironments affect cell states by comparing the different seeding sites I mentioned earlier, namely the hindbrain ventricle, the paravidulum space, and the caudal hematopoietic tissue. So when we look at some representative 3D renderings within a single seeding site, there's still some heterogeneity within the seeding site, but despite the heterogeneity, we're able to see some nice tissue-specific specialization of cell states. So this is the same PCA plot I showed you earlier, just this time represented on these specific samples as a bag plot, so we can visualize the distribution of the data. And because we know the relative contribution of specific geometric features, we can have some sort of intuition of what these axes mean in terms of cell shape. So the CHT, the cells in the CHT tend to be much more rounded compared to the PVS cells that tend to be much more elongated. And the cells in the hindbrain ventricle were more elongated compared to the CHT and distinct from both other groups. And initially, this was counterintuitive to us because the HBV was essentially our mechanically softest environment. It's essentially a fluid-filled compartment. So we thought the cells would be more rounded. So we decided to look a little bit more closely, and what we observed was a distinct cellular architecture for the hindbrain ventricle that wasn't present in other seeding sites. And that's the formation of these pseudo-rosette-like structures with cell bodies pointing outwards and protrusions pointing inwards towards a central point. And this was really interesting because it was reminiscent of a phenomenon observed in about 10% of Ewing sarcoma patients, which pathologists classify as pseudo-rosettes, so these rings of nuclei with a hollow space in the middle. And we were also able, I don't know if you see my pointer, but we were also able to see these rings, these pseudo-rosette-like structures in our hindbrain ventricle seeding sites, but not in the other seeding sites. So this was a really exciting observation for me because it made it showed remarkable parallels between the human presentation of the disease and our zebrafish model, and gave first evidence that this clinical phenomenon is actually environmentally driven. So I thought that was really interesting. So we were able to see that the different microenvironments could drive different phenotypes. We then turned to an existing model that exists in the literature in the Ewing sarcoma field, that levels of the driving oncogene themselves drive different cell states. Namely that low oncogene expressing cells are more mesenchymal-like and favor migration invasion phenotypes over proliferation. And these studies were based pretty much entirely on cell culture studies. So we wondered if this observation held true in our system, and further, if the effective oncogene level could be modulated by the different tissue microenvironments. So as I showed previously, when we looked at the unperturbed cells, we saw really nice tissue-specific specialization of cell states. But upon knockdown of the oncogene, we see this sort of indiscriminately more protrusive cell state. So that, in that sense, it phenocopied what was described previously. But further, our observation was that it had a relative loss of sensitivity to environmental cues, because the cells became more protrusive regardless of the microenvironment in which they resided. So that led us to a model where we think that the high oncogene expressing cells have high plasticity, and that they're able to respond to these different extrinsic cues, while the low oncogene expressing cells are more differentiated with a relative loss of sensitivity to the environmental cues. And it's the ability of the cells to switch between these states that could give it the survival advantage in varying stresses. So this work was published in 2022 and was a good proof of concept of the power of this system in identifying differences in cell state, how differences in cell state interact with different microenvironments. But it really relied on this, what I consider a sort of hammer on the head approach of knocking down the driving oncogene. And we wanted to see whether we could use a sort of complementary approach to find more inherent differences in cell states in Ewing sarcoma. So for that, just as a starting point, we went to single-cell transcriptomics to look for distinct Ewing sarcoma cell states with an eye specifically for surface receptors that we could target for flow cytometry to isolate and characterize distinct cell states. And that proved to be a quite productive starting point. We were able to identify a transcriptionally distinct cluster in Ewing sarcoma cell lines with lots of differentially expressed surface receptors. And one that really caught our attention is this glycoprotein called CD99. And that's because CD99 is actually routinely used as a diagnostic marker of Ewing sarcoma. It's not thought to drive the disease in any way, but it's used by clinicians as a diagnostic marker to identify the disease. So it made it a natural target to see if we could use it as to distinguish subpopulations within the cancer. And taking CD99 as a target for flow cytometry proved to be quite fruitful, because we got a really nice bimodal distribution of cell states with CD99 high and CD99 low cells, which actually after sorting were very obviously displaying very distinct morphological phenotypes. That became immediately obvious just looking at a cell culture microscope. So we have this recurring theme of a morphology representing distinct transcriptional states, like has been described in other works. I think it's important to point out that CD9, there's some degree of plasticity. The CD99 high cells, if they're isolated and then cultured again, many times they're able to shift back to become CD99 low cells again, suggesting the cells have the ability to switch between the states. So I mentioned morphologically, they're very distinct. That becomes more obvious when we look at an actin marker. The CD99 low cells tend to be more rounded with lots of filopodia, while the CD99 high cells are really flat like pancakes. And the differences are preserved also in vivo in the zebrafish xenograft model, where we see really elongated protrusions in the CD99 high cells, which can be easily quantified by differences in aspect ratio between the two groups. So what I've showed you so far in the second project is that we've discovered this distinct transcriptional state and identified CD99, which we can use as a flow cytometry target to identify these CD99 high population, which is morphologically and transcriptionally distinct. So I wondered what could be driving the CD99 high phenotype. And something that caught our attention was when we looked at these cells at the ultrastructure level. And we saw that CD99 high cells are really enriched for these membrane invaginations called caveolae. So caveolae is literally Latin for little caves. And we saw a highly enriched caveolae in the CD99 high cells. And caveolin 1, which is the protein, which is the major structural component of caveolae, is really highly enriched in this CD99 high cluster. And knockdown of CAV1 completely abrogates these structures in the CD99 high cells. We also observed that the CD99 high cells had a survival and proliferation advantage in certain environments. Here I'm showing you xenografts into the hindbrain ventricle of the zebrafish, and we can stain these xenografts for the cell death marker tunnel or proliferation marker phosphohistone 3, and we see that compared to CD99 low cells, the CD99 high cells have a massive survival advantage that's dependent on CAV1, because when we knock down CAV1, the cells no longer can survive in this environment. So these observations were really exciting to us because caveolin is a good candidate as a context-dependent regulator of signaling. So caveolae, these membrane invaginations that caveolin creates, are essentially cholesterol-rich nanodomains on the membrane that are associated with a lot of important signaling pathways in cancer, and are actually sensitive to environmental perturbations, because if you flatten the caveolae either mechanically or through cholesterol depletion, you destroy the caveolae, then you affect these signaling pathways. So that really sets caveolin 1 as a regulator of signaling that can respond to environmental cues. And when we looked at our functional proteomics assay, in addition to CAV1 being very highly upregulated in CD99, or highly overexpressed in CD99 high cells, we saw a handful of receptor tyrosine kinases that were enriched, specifically that are associated with the PI3 kinase AKT signaling pathway. And this is an important tumor cell survival pathway that's often associated with cancer progression. And indeed, in our system, we see that the active form of AKT is more enriched in the CD99 high cells and dependent on CAV1, because when we knock down CAV1, we have a loss of this active AKT signaling. So we wanted to look more upstream at the active PI3 the PI3 kinase activity. So we turned to a biosensor of active PI3 kinase, which is enriched on the membrane upon activation. When we looked between CD99 high and low cells, we didn't necessarily see a difference in enrichment of PI3 kinase on the cell surface, but when we knocked down CAV1, we had a very strong abrogation of recruitment of PI3 kinase to the cell surface. So that suggested that PI3 kinase activity at the cell membrane depends on CAV1 in these CD99 high cells. Something that we found very interesting is, though we didn't see necessarily differences in the recruitment, overall recruitment to the membrane, we wondered whether there would be different patterns of distribution of this active PI3 kinase biosensor to the membrane. But it was hard to tease out in this 2D culture experimental setup. So for that, we turned to evaluate active PI3 kinase organization in 3D, looking at cells embedded in 3D collagen. And when I did that, I observed a lot of non-uniform distributions of PI3 kinase on the cell surface, and that's non-trivial to quantify in 3D. So I leveraged a tool also developed by Hanya from the lab and published last year called cellular harmonics. And what cellular harmonics enables is quantifying the 3D surface pattern on a cell, whether it is more uniform or more polarized or more patchy, based on the calculated harmonic frequency curve. So in that way, I was able to show that actually CD99 high cells tend to have a more uniform distribution of PI3 kinase, while CD99 low cells tend to have a more polarized distribution of the signal. And it was really interesting to see that the CD99 high cells upon CAV1 knockdown actually revert to a more polarized distribution of signal. So that suggests that active PI3 kinase organization in CD99 high cells is CAV1 dependent. So that led us to a model. Yes, that led us to a model where we think caveolin-1 is regulating signaling through organization or stabilization of signaling factors on the cell surface. And in cells that don't have CAV1, the ability to organize signaling in this way is lost. So to summarize the second project, I showed you that CD99 high cells have distinct morphology, expression profiles, and PI3 kinase signaling organization. The CD99 high cells have a survival advantage in the zebrafish hindbrain that's CAV1 dependent, and through data I didn't have time to show you, also in the mouse bone cavity. And this organization of PI3 kinase signaling depends on caveolin-1. So going back to my intro slide, I talked about how these cell biological processes can really be the really drive response to microenvironment at these short time scales. And I think these imaging-based approaches I presented today, leveraging advantages in zebrafish and 3D models, can really provide new insights into these aspects of cell adaptation. So I'll end there. All my work is super collaborative, and I'm very grateful to be in a very collaborative group. My mentors are Gaudin Stanizar and Jim Amatruda at Children's Hospital of Los Angeles. I want to point out also my PhD mentor, Benny Shila, who passed away earlier this year and was very instrumental in shaping myself as a scientist and science in general. I want to thank my funding sources. And thank you all for listening. I am on the job market, and if you have any suggestions on my work or feedback or any relevant opportunities for me, I'm happy to chat further. So thanks so much for your time.
All right, let's thank Dagan for a great talk. So if you have any questions, you can go ahead and unmute yourself and ask, or paste your questions in the chat and I can read it out. Okay, so okay, maybe, yeah, so
I had a quick question. So so beautiful talk, Dagan. I was just wondering, did you look into what cytoskeletal components are driving the change in shape? I mean, the assumption is that it's actin, but it doesn't necessarily have to be.
Yeah, nice to meet you, Ashok. I've been following your work, and I'm glad to interact with you. Yeah, I think that's always a tricky question, because if you start to perturb the actin, of course you're going to affect the cell shape. So we have some people in the lab that work a little bit about that are starting to work about specific actin nucleators and how they could have different roles. We haven't reached a level where we can do that yet in 3D, spatiotemporally. I think that's interesting. I could start looking at specific actin nucleators and how inhibiting them might affect the cell shape in different ways. That would be an interesting project, and especially if I can, the zebrafish is actually amenable to drug perturbations. I can add a lot of drugs just into the water and see how they affect They actually penetrate quite well, so I could do those kinds of experiments in vivo. So
Great, thank
that's a
you.
great suggestion. Thanks.
So maybe I have a question for Dagan. So I think it's wonderful you're using the zebrafish as a model to like test how microenvironment impacts the phenotypes of the cells. So, but one question is ultimately, I think you would like to generalize your conclusion to what's actually happening in the tumor microenvironment. So, do you know, for example, how does like different tissues in the zebrafish kind of compare to the tumor microenvironment in human?
Yeah, I think that's a super important question. I showed the I think characterizing the microenvironment um, more clearly in the zebrafish model, so we could address that more directly is definitely a goal of my future lab. We have the ability to image the ECM and environment in really high resolution, which I think is very exciting. But I think ultimately we can go directly from zebrafish to spatial omics samples in humans, in human sections, right? I think that's the closest approximation to try to see if we make discoveries in our zebrafish model, and then look clinically at human samples and see if we can observe comparable distinct states. And I'm in a collaboration now with a pathologist to do that. And especially with the CD99 was an attractive target for that reason too, because these are already just routine stainings of CD99 already exist in Ewing sarcoma patients. And since it's a rare cancer, then that's a huge advantage for this kind of study. So yeah, that's ongoing.